right. Hello, everyone. Happy Valentine's Day. May any hearts you touch today be a little less slimy than the ones I was just touching. Now that we know everything about the blood, we're going to move on to the respiratory system today. Okay. So for the rest of this unit, we are going to be talking about respiration, which means that we're going to be talking about the lungs, we're going to be talking about the airways, and we're also going to be talking about gas exchange, because that's actually what respiration is. So respiration is the process of gas exchange. So we've actually already talked about a type of or a part of respiration. Okay. Specifically, cellular respiration. So when we think about respiration, we could mean any of a number of things. So internal respiration refers to the process that we've already talked about as metabolism. So internal respiration means cellular respiration. So that's that whole process where we break down glucose through all those series of steps, going through glycolysis, going through the Krebs cycle, then doing oxidative phosphorylation after we make our way passing our electrons down the electron transport chain. So that's internal respiration. That's happening at the cell level inside cells. So what we're really going to be focusing on now is called external respiration. Now, there's still a number of things in this bucket for external respiration. So when we think external respiration, we're thinking external to the cell rather than external to your body necessarily. So when we think about external respiration, we're going to start by thinking about the process of breathing, which we're going to refer to as pulmonary ventilation. So ventilation is when you feel your chest going up and down as you breathe air in and out. So that's part of external respiration, but it's not the only part. So also included in external respiration in this big category is the process by which we get exchange of gas between the lungs and the blood and the lungs and the air. The way we carry gases around in the blood. So hemoglobin's going to come back for sure. Those red blood cells we've talked about are going to come back. And then also the process of exchanging that oxygen and carbon dioxide between the blood and the body tissues. So external respiration is anything kind of external to the cells. Internal respiration is basically metabolism, basically glucose metabolism at that cellular level where we're taking in oxygen and producing carbon dioxide as we break down glucose. So today, we're going to start by thinking about the respiratory tract and some of the big organs and parts of the organ system and some of their cellular structure that we're going to need to understand in order to understand all these processes involved in external respiration. So we're going to start by thinking about the upper airways and the respiratory tract and some of these structures in the thoracic cavity. So the chest area bounded by your rib cage and then bounded by your diaphragm on the bottom. This is not going to happen again. Okay. So when we're thinking about our air passages, we're thinking about the upper airways as our passages in the head and the neck. So before we drop down into the thorax. So when you breathe in air, you can breathe it in through your nose. So you can breathe it through your nasal cavities. You can breathe air in through your mouth. So you can breathe it through your oral cavity. And you can see that these two structures come together at the back. All right, so your nose, your nasal cavity, and your mouth, your oral cavity, lead to this common area behind called the pharynx, okay? So sometimes in lab, you'll see that we divide this into a nasopharynx if it's more behind your nose, oropharynx if it's more behind your mouth, but this is the pharynx. Okay. Now, the pharynx, because it's connected to your mouth, is going to contain air, but also it's going to contain like food and water, everything you're eating and everything you're ingesting. So this is a kind of common pathway still at this point when we are up here in the head and in the neck. Where we start to see a division between our passageway into the GI system versus our passageway into these respiratory structures in the thorax is when we hit this split between the larynx anteriorly, so forward here, and the esophagus posteriorly. So that's going to go down your throat, into your stomach, into your gut tube more generally, posteriorly. Okay. So we can see that the division here is a structure called the epiglottis. Okay. Now, as we pass past the epiglottis through the larynx, so basically the larynx is also your voice box, essentially, we are going to pass down through two areas. So we're going to pass through a region we consider the conducting zone, and then we're going to go into an area we consider the respiratory zone, where the actual respiration is happening, the gas exchange is happening. So I want you to take a look at the chart that comes next in your slides about the parts of the conducting zone versus the respiratory zone. And I want you to start to kind of piece some of this out. So I want you to pick out where you can see cartilage in these structures. Okay, let's take a look. Okay, yeah. So cartilage is going to be found in the conducting zone, but not all of it. We're not going to see cartilage anymore when we travel down into the respiratory zone. We can see that we have some differences in general between these zones. So big picture, when we're thinking conducting zone versus respiratory zone, our conducting zone, what we're conducting is air. So we're going to, in general, have larger pathways for moving air. When we get down to the respiratory zone, we're going to be exchanging gas into the blood. So from the air in your lungs into the blood. So we're going to want really narrow passageways with a bunch of surface area with thin little walls where that gas exchange can happen. So you can see some of those features kind of map onto some of the things tracked here. Okay. So we said that the pharynx splits into the larynx anteriorly, going into your conducting zone. So here for our conducting zone, we're going from the larynx, the voice box, into the trachea. So this is where you can see, even in the image, C-shaped cartilages beginning. We can then see that we split out to the right and left lungs through primary bronchi. You can see there's still cartilage on here. Secondary bronchi go to each of the lobes of the lungs. We can see that cartilage there. Tertiary and smaller bronchi still have cartilage. You can see that blue cartilage here. And some of the smallest structures in the conducting zone lose their cartilage at this point, but they're still not quite as small as the structures of the respiratory zone, where we're going to start to see actual gas exchange happening. Okay. So in the conducting zone, walking through those a little more formally. So up at the top of the conducting zone, we've come from the pharynx. You have a flap covering over the larynx as you enter it from the pharynx. So like, if you think about swallowing, hopefully you can feel that you can't like breathe while you are swallowing food. That's the job of the epiglottis. It's this little kind of trap door that covers the opening to the larynx. The opening to the larynx is formally referred to as the glottis. Okay. So when you swallow, the epiglottis closes over your larynx so you don't get food down your windpipe. You don't get food in your larynx. But when you're breathing, it's open so that you can get air into the larynx, going through the glottis, passing your vocal folds, and entering the trachea. Okay. So the larynx has some cartilages that you'll see in lab forming the structure of that. They're really for anchoring a bunch of the muscles that allow you to speak. As we move into the trachea, we start to see a distinct kind of shape to our cartilages, and they're C-shaped. So from the front, right, you can see that it looks like we have rings all the way around the trachea. But we actually have our esophagus with the food that you've swallowed running behind the trachea. So there's a gap at the back of that cartilage so that when you're not breathing and you're swallowing, if you swallow a big ball of food, it can kind of use that space too. There's a flexible kind of muscular wall at the posterior portion of the trachea. Okay. So we're going to pass our air through the trachea until we split here to each of our lungs. Okay. So we split to each of our lungs, a structure called the carina here, not listed in your slides. And we're passing into our primary bronchi. So primary bronchi are the first split off of the trachea. One goes to your right lung. That's my left. One goes to your right lung, one goes to your left lung. You'll see in lab that your lungs have lobes to them. So on the left, because your heart is on the left, you only have 2 lobes, whereas on the right, where you have a little more space, you have 3 lobes. Okay? So when we think about secondary bronchi, we're going to those different lobes. So you'll have 3 secondary bronchi on the right, 1 going to each of the 3 lobes. We'll have 2 secondary bronchi on the left, going to the 2 lobes in that left lung. Tertiary bronchi start to go to different segments of the lungs. So segments aren't visible when you look at the lungs in lab, but like physiologically and developmentally, your lung has a lot of different segments. So you'd have tertiary bronchi splitting to those different segments. Then we have splitting and splitting and splitting. So as we get smaller, we travel to the tertiary bronchi, to the super scientific here, smaller bronchi. Once we lose our cartilage and hit a diameter that's less than about 1 millimeter, you're now in a bronchiole. So just like our arterioles were smaller than arteries, bronchioles are smaller than bronchi, but they're attached to each other. Okay? Your bronchioles are still part of your conducting zone, even though they don't have that cartilage, they're still pretty wide. They continue splitting, giving us the terminal bronchioles. The terminal bronchioles are the last step of your conducting zone before we hit the respiratory zone. Okay. Now, before we talk about the structures of the respiratory zone, let's think about what you'd see if you, say, put a little bit of tissue from the conducting zone on a slide and looked at it under a microscope. Okay. So purposes of the conducting zone, as an air passageway, we don't have respiration, gas exchange happening here in the conducting zone. So we sometimes do think of this as kind of dead space. So you're holding some air, but you're not actually absorbing any oxygen in that air or putting carbon dioxide into that air while it's in the conducting zone. It's about 150 milliliters of volume of air that'll be in the conducting zone at any minute. We're pointing that out now because Later on, we'll be doing calculations and thinking about how much fresh oxygen you're getting as you breathe. And so this comes back into play. So you're holding some air in this dead space. And as you're doing that, super relevant to us in winter here, you're increasing the air temperature, right? So even as you're breathing into your nose, but certainly as it's moving through the passageways in your neck and then descending down into your chest, you're trying to make that air basically a temperature that's going to be nice for your lung tissue. Lung tissue is kind of fragile. Lung tissue is delicate. It's really thin. It's designed for that gas exchange process, swapping the oxygen and carbon dioxide. So we want to make the air nice and predictable and warm and humid. So as the air is passing through the conducting zone, it's warming up to your body temperature. And it's getting wetter. We're humidifying it. These structures are lined basically with mucous membranes. So there's fluid there a little bit on the surface that's helping us humidify the air. If we look a little deeper, right? So zooming in even more, if we look at the epithelium of the conducting zone, so that lining that's separating the air in the conducting zone from the tissue of your body and from your blood, we're going to notice a couple of important types of cells. Okay. So in the conducting zone, we have these two types of cells called goblet cells and ciliated cells. And they're important for you and your life and your body, in part because they're helping make sure that you don't get like little particles stuck in your lungs, that hopefully you don't have like pathogens making it down into the tissue of your lungs. So they're kind of defending you both from physical damage, as well as a little bit potentially from some biological invaders. Okay, so the way they do that is through something called the mucus escalator. Not an escalator I would like to be on, but an escalator that we should all be very glad that we have in our throats. Okay. So what the mucus escalator is we have these goblet cells that are secreting mucus, right? So we're making this kind of sticky lining on the epithelium that's lining our conducting zone. And then we have these ciliated cells, and cilia are basically like little hairs, little fingers. And the ciliated cells are just moving like this, moving stuff up towards your mouth, right? So we are moving mucus up towards our mouth kind of all the time, right? What that mucus is supposed to do, it's supposed to catch particles that have come in through the air. They're going through the conducting zone, headed towards your lungs. We would like to trap them in the mucus and then pull them up to your mouth so you can like cough them out, spit them out, so they don't end up in your lung tissue, right? So this is their role. So you can see them here, those goblet cells producing mucus in this little yellow zone. And here you can see the ciliated cells with their hair-like cilia facing where the air would be traveling. So this would be the lumen, so the like open part of, let's say, your trachea. Here's what it would look like on a histological slide. So we can see these glands and such in the tissue, right? We'd be producing mucus. Here we can see the cilia of the ciliated cells right on the surface. And we can tell this is the lumen of the trachea on the right. The lumen of the trachea being the inside of the passageway, partially because we can see cartilage all the way over here on the left. We know that's the outside. So a fun sort of relatively common correlation that we can make in order to understand why we care about these ciliated cells and these goblet cells and the mucus escalator is if you think about smokers, how long-term smokers will develop a smoker's cough sometimes. Part of the reason for that is that their mucus escalator has kind of stopped working. So smoke can paralyze those ciliated cells. But you still don't want gunk collecting down in your lungs, right? So the only way then to get gunk out of your lungs is to try to forcefully push lots of air out of your lungs and then out through the conducting zone and out your mouth. And so you have to actively use musculature to do that. So your diaphragm is working, your muscles attached to your rib cage are working to forcefully expel air to kind of make up for the fact that this mucus escalator isn't working. Okay, so that's the conducting zone. Hopefully pretty simple. We got a series of tubes. We've got the mucus escalator trying to make sure that our air is clean. In general, we're warming up air, we're humidifying air. Then we hit our respiratory zone. So the respiratory bronchioles, the first part of our respiratory zone, they're following the terminal bronchioles of the conducting zone. And what they can do is start to do some gas exchange. Okay. So they're getting small here. They're less than 0.5 millimeters in diameter, and they're small enough, and their membranes, their linings are thin enough that we can allow gases to cross between the air and the blood that's lying right next to all of these divisions. Okay. So when we have gas exchange happening, reminder, the gases that we're interested in, oxygen and carbon dioxide, they're small, they're nonpolar, they can cross cell membranes relatively easily. Okay, so all this gas exchange that we talk about throughout this unit is happening passively. So this is a passive transport, and specifically, it's just diffusion, simple diffusion happening here. We're not going to need channels and transporters or membrane proteins to make this work, right? It's just little thin walls, simple diffusion happening across the wall. Okay. So the respiratory bronchioles start this process of gas exchange, and that continues as we dive even deeper into the lung. So the main structure that we think about when we think about the majority of our gas exchange in the respiratory zone is happening in these little yellow pieces. So these little yellow circles here, these little clusters of grapes, those are alveoli. So alveoli are those little grape-like structures, and they're all connected to each other. So we have alveolar ducts leading into them. We have alveolar sacs surrounding them. But all of this structure is really designed to maximize surface area so that we have lots and lots of space for gas exchange. Okay. So here we can see zoomed in a little further on some of those structures, and we're going to keep zooming and zooming. All right, so here's the terminal bronchiole coming down from the conducting zone. This respiratory bronchiole, you can see is starting to even pick up like a couple of alveoli kind of sitting on it here. That's part of why it can do the gas exchange. But the main part of the respiratory zone is here as we start to pass into an alveolar duct, leading us to an alveolar sac, which is really just a group of alveoli all clustered together. And now we're going to zoom in on that alveolar sac. Okay. So each of these little circles is an individual alveolus, plural is alveoli. This is where our gas exchange is really happening. You have lots and lots of alveoli, about 300 million of them. And if you were to peel them out of your lungs and spread them all flat, you'd get something about the size of a tennis court. So that's the scale of the membrane that we're working with as we're doing gas exchange in the lungs. Now, if you look at the alveoli and this alveolar sac, you can see that we have capillaries sitting all over the top. Now, we have a really close association between the alveoli, which are going to be filled with air, and the capillaries sitting on top, which, as we know, are filled with blood, right? And that's really important because we need the gases to go from the air to the blood. And if we're talking about oxygen, and then if we're talking about carbon dioxide, we need to be going from that blood into the alveoli so that we can breathe out carbon dioxide. So it's really good, really important that we have this dense capillary network here. You'll notice we also have connections between the alveoli in this alveolar sac. So these little alveolar pores, these little dots, they're connecting neighboring alveoli. Okay. So now that we're in the respiratory zone, we're in the alveolar sac, we're in the alveoli. We are also going to talk about some of the cells involved here in making up the walls of the alveoli. Okay. These numbers are relatively helpful, unlike the numbers on all our different clotting factors last time. Okay, so type 1 alveolar cells are the ones that are the bulk of this, making up the wall of the alveolus. So the main structure of the alveolus is made up of these type 1 alveolar cells. They're just a single layer of epithelial cells making this kind of thin wall of the alveolus that touches the thin wall of the capillaries. If you remember back to the last unit, capillaries are really just one thin layer of cells too, right? So we've got this really thin layer as we're going from air in the alveoli, crossing epithelial cells, the single layer making up the wall of the alveolus. And then all we have to do is cross the single layer of the capillary wall to get into the blood. Okay. So the type 1 alveolar cells are most of them making the wall of the alveolus, but these other cells have important functions as well. So type 2 alveolar cells secrete something called surfactant. So a surfactant is something that acts to break up water tension. So you can think of it kind of getting in there like a little like soap or something, mixing in. And the reason this is important in the lungs and in the alveoli is it's going to prevent them from collapsing. So the way it does that is basically if you have alveoli get small, right? Water tension, water pulls towards itself. So you might have a tendency for the walls of the alveoli to kind of smack towards each other and collapse an alveolus. We don't want that to happen, so we need surfactant. So type 2 alveolar cells, still important functionally. We also have macrophages here. So we mentioned macrophages were one of our types of leukocytes, one of our types of white blood cells. Right? So we've got some alveolar macrophages here in the lungs, continuing to help defend our tissue. Right? So we had the mucus escalator trying to catch big pathogens and particulates and get those out your mouth before we even get into the respiratory zone. Now we've got the alveolar macrophages. They're going to try to eat anything that made its way down, made its way past that mucus escalator. So what we're seeing here is that association between the capillaries and the arterial blood flow here and the airways into the respiratory zone. So here you can see the trachea, and everything in yellow here would be our bronchi, and then into our bronchioles, and then into alveolar sacs. And here you see the pulmonary artery leading into many, many branches that lie right alongside all those airways in the lungs. And if we zoom way in, this is what we would see. So here we see those capillaries sitting right next to little alveoli here. So really close association. Okay. So here we're zooming in cartoon form on those types of cells that we just mentioned, right? Okay. So the type 1 cells, the type 1 alveolar cells, the ones that make up the main part of the wall, are these yellow ones, right? Those type 2 alveolar cells that are secreting surfactant are these green ones. So you can see we have fewer of them, but they're interspersed throughout the alveoli to make sure that they're secreting this surfactant so our alveoli don't collapse. And here you can see a couple of those macrophages here, ready to catch any pathogens that make their way down this far into the respiratory zone. And then you see the capillaries all the way around. Okay. Zooming in even further, we've zoomed in here on one of those yellow cells, so one of those type 1 alveolar cells, and its neighboring capillary. Okay, so here we've got that neighboring capillary. So we can see that when we're thinking about the membrane that we have to cross in order to get from the air to the blood, or in the opposite direction, from the blood into the air. You can think of that as our respiratory membrane across which gas exchange is happening, right? So that's the respiratory membrane where we're exchanging our gases. So you can see how thin it is, right? We've just got one epithelial cell. We've got a little basement membrane that's just basically acting as glue here, sticking us to our capillary endothelium. Okay, so a reminder way back from A&P1, it's epithelium if it touches some external environment, right? So air outside counts as external environment, but air in your lungs is still external environment because you haven't processed it yet, you haven't absorbed it yet. So that's why the alveolar cells are epithelium. Endothelium lines cavities inside your body, separating different parts of your internal environment. So that endothelial layer of the capillaries is endothelial because we're touching the blood, which is an internal part of your body. But functionally, they work very similarly here. Right? So we're just crossing alveolar epithelium, teeny tiny basement membrane, which we don't really have to think about when we think about what might block gas exchange. And then we're going through the capillary endothelium. So this is about 0.2 micrometers thick, so really thin, which is great. And here is an SEM photo looking at those alveoli as well. So scanning electron microscope. At least that's my assumption. I feel like that's always what these craggy kind of black and white photos are. Okay. Zooming out, looking at some larger structures. So when we think about ventilation, so pulmonary ventilation, physically moving air in and out of the lungs, we're going to be thinking about the muscles you need to use. We're going to be thinking about bones you need to move. What we're going to be trying to do overall to drive that ventilation is we're trying to change pressures in the lung. Okay, so we need to physically move large structures to do that. So when we think about the chest wall, right, so the wall of the thoracic cavity, we want this to be airtight, except for that conducting zone going in, right? We don't want any gaps between our ribs, letting air in and out across the chest wall, right? So we're going to have a bony skeleton that forms the physical structure. So we're going to have our rib cage, so our ribs coming around, sternum in front, thoracic vertebrae in back, creating this cage protecting the lungs. And between those ribs, we're going to have our intercostal muscles. Particularly important for us will be the internal and external intercostal muscles. So you'll see these are like little thin layers of muscles with opposite fiber directions you'll see in lab. Okay. So those are between your ribs. They're able to pull on the ribs, which means they're going to enable your thoracic cavity to change shape. And then we're going to have a diaphragm. So the diaphragm is like a flat sheet of muscle. I mentioned it when we were talking about the respiratory pump for moving blood through veins. So it's coming back and much more important now. I'm going to draw that. All right. So rib cage, and my little rib cage, bad rib cage, diaphragm, sealing off the bottom of the rib cage and separating us from the abdomen and the abdominal contents. Okay. So those are our big structures. We got bones, we got muscles, physically protecting the space and allowing it to move and change shape, and therefore change volume and change pressure. We also have linings. So we have membranes lining the lungs. We have a membrane lining the chest wall. They're kind of continuous with each other. So if you look at the root of a lung, so where the lung attaches to the bronchi and the trachea and all those structures of the conducting zone and airways. You'd see a little kind of corner, but you see pleura. So parietal pleura, you'll see line the inside of this wall. I'll draw. Okay. Let me draw one lung. Oh, we can have two lungs. Okay. Okay. So if we have a lung here and a lung here, you'll have parietal pleura lining the walls. And you'll have visceral pleura lining the outside of the lungs. So overall, we're making like a bag. So when it says pleural sac here, this is very not to scale, right? But this wall part and this lung part communicate with each other here. They're attached to each other. So we have this pleural sac with an intrapleural space that I'm just kind of cross-hatching right now. Okay. All right. This is good because it means that as your lungs inflate and deflate, there's no like friction as they rub against ribs and muscles and stuff. This pleural sac is nice and slimy. So we've got this low friction. So hopefully not creating any extra forces on the lungs, making it harder to expand. So this intrapleural space that I was trying to fill in with blue there is filled with a little bit of intrapleural fluid, about 15 milliliters. Now that intrapleural fluid also helps the two sides of the pleural sac, right? So this parietal part and this visceral part to kind of stick to each other, right? Because water has surface tension. I mentioned that's why we need surfactant in the alveoli, because we don't want alveoli to have their walls sticking to each other. But you do kind of want the surface of the lung to follow along with the inner surface of the thoracic wall as you use muscles to change its shape so that we can kind of change the shape of the lungs, change pressures, and move air. So here's a prettier drawing of those structures, okay? So here we can see ribs, sternum, right? We don't have to draw those on again. Hopefully we're all familiar. Here we can see the intercostal muscles connecting our ribs to each other. So we'd have external, and then we'd have another layer inside our internal. Okay. Here is our diaphragm sealing over the bottom. So you can see it here as well. It's forming a flat sheet. You can see those muscle fibers running across, right? So the intercostal muscles and the diaphragm are going to be forcing the movement by contracting. Here in purple-ish, you can see that pleural sac around the lungs with a part that touches the chest wall and a part that's just on the surface of the lung, okay? And you can see that there's a little bit of space here in between, which is where the fluid would lie. That 15 milliliters in that intrapleural space, that intrapleural fluid, kind of holding these two things together. Okay. We are going to think about the forces for ventilation a little bit now. Okay. So when we think about pulmonary ventilation, we're talking about really the bulk flow of air. So physically moving a bunch of air into the lung and then pushing that air out of the lung. Okay. So this is going to work similarly to the bulk flow of blood that we talked about in our last unit. Okay. So trying to make this analogy, trying to pull it together for ourselves. If we've got an analogy between the forces allowing the flow of blood and the forces involved in ventilation, which is the bulk flow of air. What do you think is going to be driving that ventilation? What do you think is going to be the big force that allows for a lot of movement here when we're thinking about a bunch of air? Take a look at our vote. Okay, cool. All right, so almost everybody got that we're thinking about a pressure difference, which is great because yes, this is all driven by our flow rule, right? So bulk flow is still just bulk flow, even though it's air now, not blood. So we have a pressure difference and we're gonna have resistance, okay? And when we're thinking about this pressure difference, when we're thinking about ventilation, what we're trying to do is move air. If you're breathing in, inhaling, we're trying to move air from the external environment into the thorax through the conducting zone initially. Okay. So it's going to be that pressure difference between the air that's outside your body and the air that is moving into the lungs that's driving that flow. Between the thorax and the abdomen was what we were thinking about when we were thinking about veins and the movement of blood back to the heart. But we're not actually going to be moving air into the abdomen, hopefully. Okay. So our air is moving through bulk flow, same equation as last time. Flow is a pressure difference combated by some resistance. So just like blood, just like every gradient we've seen in this course and in last semester, air is going to be moving down a gradient. So air is going to move from a place where you have high pressure to a place where you have low pressure, right? And you can kind of understand that physically if you think about what it's like to puff a lot of air out forcefully, right? Like if you go, you can feel that what you've had to do is build pressure inside your body, build it so much that it's more than the pressure outside you so that you can move that air out, right? So that's air moving down its pressure gradient from where we have higher pressure to where we have lower pressure. Okay. So the difference between inspiration, so inspiration is breathing in, and expiration is breathing out when you're exhaling, is going to be which of those two pressures is bigger, the pressure in the lung or the pressure of the atmosphere around you, okay? So we're not going to be able to change atmospheric pressure, right? Like you can't really change the pressure of the air in this room. Like you'd have to do a lot of engineering to try to figure out how to do that. You're not doing that breath by breath, right? But what you can change is the pressure in your lungs, and you can change that pretty easily. So when we inspire, when we breathe in, when we inhale, what we're doing is creating pressure in our lungs that's lower than our atmospheric pressure. So we're trying to create like a vacuum. Okay. So for inspiration, here are my lungs, right? All we're trying to do is create low pressure relative to a higher atmospheric pressure. And that is going to naturally draw air into the lungs as it tries to find that low pressure environment because everything is trying to get to equilibrium, right? Everything's trying to even itself out. So that's our inspiration. And our expiration is just the opposite, right? So we are going to try, when we expire, when we exhale, we're going to be building pressure inside the lung, right? So we're creating a high pressure system here. And now that that's high, this is all relative. So it's going to be relatively high compared to atmospheric pressure. And that's going to force air out of the lungs, still just going down that high to low gradient. Okay. So we're pulling air in during inspiration, using that low pressure in the lungs. We're pushing air out during expiration as we create a high pressure in the lungs. We are also going to have a kind of even point, right? In order to do that, right? At some point we have crossed atmospheric pressure, right? So atmospheric pressure is going to be stable. To inhale, we're dropping pressure in the lungs below it. To exhale, we're pulling our pressure in the lungs above it, which means there's a point at which we're even. There's a point where we crossed that atmospheric pressure. The volume of lungs that's in your air between breaths, as you are neither moving air in nor moving air out, it's going to be an amount called your functional residual capacity. Later on, we'll come back to lung volumes and lung capacities and what they mean for us, right? Because those are going to be things that you could like measure in a patient to think about, are their lungs functioning appropriately? Are they getting enough oxygen? Are they clearing enough carbon dioxide? And are they doing it fast enough? Okay. So what we are going to be thinking about as we examine inspiration and expiration are a variety of pulmonary pressures. Okay. So for these pulmonary pressures, we've already talked about the two most important ones. Okay. So when we go through questions about pulmonary ventilation and I ask you, is this person breathing in or is this person breathing out, right? Are we inhaling, inspiring, or are we exhaling, expiring? All you actually have to look at to answer that question are these first two. Okay. So when I was just saying that we're moving from high to low down a gradient, whether that's drawing air in or pushing air out, we're comparing to our atmospheric pressure. So that's what this PATM is. P for pressure, ATM for atmosphere. And the pressure we really care about most when we're thinking about the lungs, when I said lung pressure, right? What I mean is pressure in the alveoli. That's the most important one because that's where our respiration is actually happening. That's where gas exchange is happening. So the alveoli in those little alveolar sacs, those little baggies of air, our PALV, that's our intraalveolar pressure. And so that's what's going to be driving, are we pulling air into the alveoli or are we pushing air out of the alveoli, breathing out, sending it to the external environment? The other two pressures are important for understanding a little bit of how and why this happens, but those top two are the really important ones that you're really going to be focusing on. Okay, so we'll pick up here next time and do some comparisons of pressures, etc.